Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing the remaining three type of crosses where we have two genes and the cross involve two genes where they may have one allele each, two allele each, or they may have more than those. So let's start with this. So the discussion that I'm about to have start here is independent assortment of genes and alleles. So what do you mean by independent assortment of genes and alleles? We have discussed this in detail in my video regarding meiosis. So how independent assortment of alleles and genes occur. So for example, if there is an individual cell which is having capital A, small a, capital D, small d, this individual, where is this capital A, small a located? The capital A, small a is located on a homologous pair. So this is a homologous pair. For example, there is capital A here, and there's a small a on the homologous pair, the other chromosome. The same individual may have another gene capital D, small d being their allele on another homologous chromosome. For example, this is yet another homologous chromosome, and there's this gene capital D allele being there and a small d being there. So this is what I'm representing. This is one homologous chromosome having capital D, small d. This is the other homologous chromosome pair having capital A, small a. So the overall genotype of this organism is capital A, small a, capital D, small d. Knowing that at least one capital A is there, at least one capital D is there, so the phenotype will be denoted or will be governed by these dominant alleles, one being dominant over the small a and the other being dominant over the small d. So more of that of the phenotype later on, but yes, this is condition called dihybrid. What do you mean by dihybrid? Dihybrid means the person or the individual is heterozygous at both gene loci. So this is heterozygous at both gene loci. So the gene loci means the location of gene on the chromosome. So this individual is heterozygous at both gene loci. Let's move on. So when the cell undergoes prophase one, we know that the chromosomes start to align. The bivalents pair up. What do you mean by the bivalents pair up? The bivalents form the homologous pair up, line up by side by side. So this is a one bivalve, the homologous chromosome pair, the other comes together. And when they align on the equator of the cell, the alignment can be independent of each other, which is called the independent assortment of chromosomes. So they may line up in such a way that the one dominant and one recessive allele chromosome faces one another, and so on and so forth. Or they may align in themselves that both recessive allele chromosome are at the top and both dominant allele chromosome at the bottom. So when this occurs, this is called independent assortment of chromosome. It is because of the random alignment of the bivalents along the equator of the spin of the cell. So when they will separate after metaphase one, this small a and small d will be pulled up. So you will end up having a cell of small a and capital D, and these capital A, small d will be pulled down. So you will have another cell having one dominant and one recessive allele. Similarly, this small a, small d goes up. So a cell comes like small a and small d, and this comes down. So this will become a cell of capital A, capital D. So this is independent assortment. You have four different type of gamete being produced, and all four gametes will be produced in equal quantity. There's a chance of one dominant allele coming with the other recessive allele. There's a chance of one recessive coming with the other dominant. There's a chance of both dominant alleles ending up in the same gamete. There's a chance of one recessive and the other recessive being in the same gamete. So four different type of gametes will form, and these four different type of gametes will be all in equal proportion. This is what will happen by the end of meiosis. Now, we know this these two letters, they represent your bivalent, this represent the heterozygous condition. So if you make gametes of these are the four different type of gametes that will be produced. So how do you make gametes? When you add gametes, you just write one capital letter from the first set and the second capital letter together. This forms the gamete having both the dominant alleles together. You make another gamete with one dominant of this, and the other is recessive here. So this is the second type of gamete. The third gamete here we're having a recessive allele with the dominant, and the fourth is having recessive and recessive together. All four different gametes are possible. The same thing can be carried out for another set. For example, if you were given a trait where these symbols are being used, again you will have four different type of gamete being produced. Now, if the organism was not a dihybrid organism, rather a purebred dominant, or having a homozygous dominant at both gene loci, or homozygous recessive at both gene loci, the individual again produce four type of gametes, but all these four type of gametes in the first person, this one, would all be same. All will have the dominant combination of G and B together, whereas an individual who is pure but recessive will end up producing only the single type of gamete, again four gametes and both are being produced, but the four gametes are not different. In the dihybrid condition, this is an individual where four different type of gametes are being produced. Similarly, if we take an example of, let's suppose an individual is capital G, or capital G. Oh, okay, let me just take this. For example, the individual is capital G and this small g, and the person is having capital B, small b. In this case, again, you will have four different type of gametes as already mentioned. Now let's just change this as a condition. For instance, I change this one as small b, small b. Now, in that case, what type of gamete will produce? The individual will have one gamete where you will have capital G with a small b, the other will be having small g with a capital B, and that's it. Again, both other gametes will again be the same, capital G with a small b and a small g with a capital B, sorry, small b. So this case again shows that two gametes will be similar and these two gametes will be similar. So four different gametes are not being produced. Yes, four gametes are produced, but among these four gametes, two gametes are alike and the remaining two gametes are alike as well. So you should know how to make gametes from a given set of letters. That's the first thing that you should be knowing before we start the cross. Now, when we are doing the crosses, what do we do? For instance, this is the rule that I explained that you follow whenever you are doing crosses. So let's take an example. For example, if an individual who is homozygous dominant at both the gene loci and homozygous recessive at both gene loci, if they are being crossed together, they will end up producing F1 offspring, which will be 100% hybrid of this condition. Now, this F1 offspring being hybrid, this hybrid condition will have heterozygous genes, heterozygous at both. This is called as the F1 offspring. Now, this F1 offspring will be expressing the features of capital A and capital D very much similar to one of the parent and not the other because the recessive traits were being expressed there. Now, if we allow this F1 offspring to intercross, F1, F1 together, if both F1, F1 offsprings are crossed together, we get a ratio of 9331. This 9331 ratio is of recombinants. You have two recombinant being produced where you will have feature of both parents being expressed in one individual, and 9 and 1 will represent the parental phenotype. These are four phenotypes being produced in the F2 generation. And among these four phenotypes, the ratio will be 9331. Then let's suppose if we were to cross the F1 offspring with the purebred recessive. In this case, again, you will have four different phenotypes, but they will all be in the ratio of 1 all. Again, you will have two recombinants here. What are two recombinants? We'll be discussing them with the examples as we go forward. So if independent assortment is being followed, independent assortment simply means that four different type of gametes were produced. There was independent alignment of chromosome occurring at the equator, and this ended up producing four different type of gametes. So these ratios will be followed. 9331 will be followed when you have two dihybrid being crossed together, where you will have two recombinant being produced. And if you cross an F1 offspring dihybrid with a purebred recessive, you will have four phenotypes all in equal proportion. Now, this brings us to the fifth type of cross as far as your syllabus, and the second cross in fact, the first cross of two genes being considered together. This is called a dihybrid cross. What's a dihybrid cross? It's a cross involving two genes controlling two different traits at the same time. So let's take the example of a plant having capital A for purple stem, small a for green stem. We know if it's a purple stem or individual, the capital A allele is enough for this expression. We can mention capital A dash as purple stem. This dash could be capital A, or it could be capital A dash, which means capital A small a. So both these will be purple. Similarly, small a is considered to be green stem here. The information is already provided to you. Gene for leaf shape. For example, the leaf shape is on a separate chromosome. Capital D for cut leaf, small d for potato leaf. So these are the combination of capital D, small d, and capital D. Capital D both being cut leaf and small d being potato leaf. You don't have to learn or cram anything out of it, but yes, you need to know how to make the combinations of these. So these are four different phenotypes being displayed among various genotypes. These are all possible genotypes that can be there in an organism, individual organism. So a person can be purebred dominant at both gene loci, a person can be purebred recessive at both gene loci, a person can be, or individual in this case, the plant can be heterozygous at both gene loci. The individual can be heterozygous at one gene loci, but homozygous recessive at the other gene loci. The organism can be homozygous dominant and homozygous recessive for both gene loci. The person can be homozygous recessive at the first gene loci, but the heterozygous dominant for the next gene loci. So individual information will be given to you when you're asked to do the question. but you need to know what the letter represents. So at least one capital A being there in this condition, in this example, will give a purple stem, and at least one capital D will end up getting cut leaf. So similarly, if both small a's are there together, the individual will have green stem. If both small d's are together, in this case, the individual will have potato leaf shape. So there are four phenotypes: purple cut, and there's purple potato, and there's green cut, and there's green potato. So four different phenotypes can be seen. So this is how the crosses go. So if we take the first cross of purebred dominant with purebred recessive, we'll get 100% hybrid offspring. Then this is called the F1 offspring. If we cross the F1, F1 together, we get 9:3:1 ratio. If we cross F1 with purebred recessive, we get 1 all ratio. And in all these ratios, if an independent assortment is being followed between the genes of capital A small a and capital D small d, if independent assortment is occurring, it means that 9:3:1 ratio will be followed, 1:1 ratio will be followed, and there will be two recombinants in either case. So let's take this cross here. So the first cross between purebred dominant and purebred recessive, we know that in this case they will produce all the same kind of gametes. So all gametes will have dominant alleles together, and here all the gametes will be having the recessive alleles together. You get 100% offspring, which is hybrid, expressing the phenotype of the dominant parent, purple cut. So this individual was purple cut, the other was a green potato. So in this case, all should be purple cut. This should be all green potato, and we see that this purple cut feature being passed on, and the alleles for green potato being there but not able to express themselves because it's a heterozygous state. Now, if we pick the F1 offspring and cross it with the purebred recessive means that all the gametes will be recessive trait together. Now, if you cross them together, rather than making one box or two boxes, we'll make a 4 by 1 box. 4 by 1 box, four being four to represent the four kind of gametes that this individual will make, because we'll make four different kind of gametes, whereas this one will make only one kind of gamete, and all four gametes of this individual will be of the same type. So rather than drawing it as a 4 by 4 box where 16 boxes will be drawn, I'm drawing just one representative box, because all these will be the same as compared to this, because every time you cross capital AD with a small ad, you will get the same outcomes here. So I have ignored all these together, and I'm just drawing the first column of this box. So this is shown to you here in this diagram. When you cross them together, we see that two chances. First of all, we see four phenotypes here, and four phenotypes. Why? Because you have purple cut being produced, you have green potato being produced. They both are parental class, but we see a new feature: purple stem with potato leaf, and green cut with green stem with with cut leaf. So these both are new variety. These both are called the recombinants. And again, you have 1:1 all ratio, and these are new phenotypes being shown here, recombinant shown here. So this means that yes, independent assortment was occurring. Capital A small a, capital D small d, they both follow independent assortment. The offspring that have been produced here are called the F2 offspring because there's a second generation. Now, the other cross that we can do here is we can cross the F1 offspring together with another offspring, which means a dihybrid being crossed with another dihybrid. In this case, you will be drawing a 4 by 4 box, which is a 16 squares box. So in this case, you will have four type of gametes different at the top from one of the parent, and four different type of gametes from the other parent on the other side. When you cross them together, just mention them as an outcome. And wherever you have at least one capital A, that becomes purple. Wherever you have two small a's together, that becomes green, according to the information provided. Similarly, at least a single capital D will make the individual cut, and the small d's being together will make the individual potato. So if you start to cross and count them, you will see four different phenotypes being produced. And among these phenotypes, again, purple cut and green potato will both be parental classes. Similarly, purple potato and green cut will be the recombinant classes. They will be in the ratio of 9:3:3:1, a total of 16 offspring being produced in the ratio 9:3:3:1. If these ratios are followed, this means the gene AA and DD, they were following independent assortment. This takes us to the sixth type of cross. In all, we call this epitasis. In epitasis, there is interaction between loci, there is interaction between gene occurring. So epitasis is the gene interaction where one gene interferes with the expression of the other. The gene are situated at different gene loci, this suppress the genes. So the normal phenotype effect of the affected gene are not observed. The F2 generation will not follow 9:3:1 ratio of four phenotypes. How is that going about? For example, let's take an example of a gene capital A, small a produce a gouty or yellow coat color in for example in these guinea pigs or these mice. So this is there, and small a produce yellow. Capital A dash is a gouty, which means capital A is a gouty, capital A small is a gouty as well. Similarly, gene capital B small b controls pigment production, which means this combination it must be there for a color to be expressed. It is mentioned that capital B dash pigment produce, which means capital B small b will have a color, and the color could either be a gouty or yellow. But if the same animal is having small b at the gene B locus, the organism will be albino irrespective of whatever combination of A's it may have. If it's small b, it will be albino. So this gene interferes with the expression of color production. So this gives us three phenotypes, agouti, yellow, and albino. So let's move on with the cross. So what do we do? The first cross we are doing is between the purebred dominant and purebred recessive for both the gene loci. They will produce the same kind of four gametes each, and when we cross them together, you get the dihybrid offspring here, which is the F1 offspring. Now, when you cross the F1 offspring with another F1 offspring, we expect 9:3:1 ratio to be followed if it's an independent assortment, but we will not have this ratio 9:3:1. Why? Because one gene is affecting the expression of the other. So if you perform the second cross here, I would recommend that you go and just perform this cross on your own. This is the cross that I'm talking about. You take agouti and yellow, we know the genotypes from the phenotypes, and these as well. The F1 intercross is being conducted. You have a dihybrid and dihybrid with each other being crossed together. You will make a box of 4 by 4, and just start putting the cross together. You will have three phenotypes being expressed rather than four phenotypes, and the ratio will come out to be the way you do. Just keep in mind that if there are two small b's in an individual, in this case the individual will be albino, because it's already mentioned in the description that this feature will give albino. This animal cannot be yellow or agouti if small b is present together. So such an interaction is called epsitasis. The last kind of example or the content that you have to cover is called autosomal linkage. What is autosomal linkage? Autosomal means the genes are located on autosome and not the sex chromosome. Here, two genes present are on the same chromosome. They are not on the different chromosome. They are said to be linked because they are very close together. There is very less chance of recombination, which means going from one chromosome to the other when crossing over occur. The farther apart the genes are, the more chance of combination they have, the recombination they have. What will happen here? For example, the genes are located so close to another that they are of the same chromatid, they tend to stay together, forming a linkage group. For example, this is the homologous pair shown to you. Capital A and capital B are the genes, the alleles present on one chromosome, and the other homologous chromosome pair, the chromosome of the same pair, is having small a, small b. Now, if crossing over was to occur, these genes are so close together physically, capital A and capital B are so close that if capital A goes there, capital B goes there as well, and small a and small b comes here as well. So the exchange will eventually have no effect. So when they eventually do have no effect, we have only two type of gamete being produced. The gamete will have capital AB together or small ab together. So only two type of gamete will be produced. We will not have a gamete of small a with a capital B or capital A with a small b. Why? Because the crossing over did not occur. It did not occur that capital A goes there and small a comes here. So the recombination was not there. This is called linkage. So if no crossing over events occur, only two type of gametes are produced, in which the linkage group is passed on as it is into the gamete without crossing over event. The same thing goes on in both the gametes. So you have just two type of gamete being produced. Now let's suppose if the genes do cross over, there are chances that yes, even in linkage, the genes can cross over, but the ratio will not be the same. So in this case, what's happening? The linkage group breaks and you will have the segment being swapped. So capital A goes there, small a comes here. When they come, in this case, you will have a gamete of capital AB being produced. You will have a gamete of a small a B being produced. You will have a gamete of a small a with a capital B being produced and a capital A with a small a B being produced as well. But Yes, four gametes are being produced, but are these gametes in the equal proportion? No, they will not be in equal proportion. You will have more gametes which are having the linkage group intact, and very few gametes will have the linkage group broken. The ratios again will not be the same, and even if 9331 is being followed, number one 9331 ratio will not be followed. Four phenotypes will be there, but not in this ratio. Similarly, if 1:1:1 ratio is being followed, again the ratio will be there, four phenotypes will be produced, but they will not be in the same ratio, they will not be in same proportion. The recombinants which are being born because of these will be in very few quantity or very few numbers as compared to the parental class. For instance, here they have given an example of fruit fly where capital E is for the striped body and small e is for the ebony body. The ebony means black colored. Antenna shape is capital A and small a for the Aristopedia antenna. We are having the first case of both being homozygous dominant and homozygous recessive. So we are crossing individual capital E, capital E, capital A, capital A with a small e, small e, small a, small a. We know that when they cross together, all the individuals produced will have these conditions. A dominant E and a dominant A with a recessive E and a recessive A will be produced as F1 offspring. 100% hybrid offspring will be produced. This is the F1 offspring. The next we can do is cross this F1 offspring with a purebred recessive. So this is what we are doing. We took the purebred recessive and we took the this F1 offspring. So you have this capital E with a capital A, small a, cross with small e, small a, small e, small a. The way they have written them as separate is because of the linkage group. To make things easier, I'm just writing it the way that we have been writing. So what do? We have two type of gamete being produced here, and you have just one type of gamete being produced here. This is the outcome of the cross. The F2 offsprings do not show four phenotypes. The offsprings show only two phenotypes and only the parental class being produced, which means if it was a striped body together with a normal antenna, you got the same in this condition and striped and normal state in the F2 generation as well. Similarly, if Aristopedia antenna and ebony body were there together, you see the same feature being passed on in the next generation, ebony and Aristopedia. You do not see any individual being produced which is striped and Aristopedia. This one is not, this individual is just not produced. Similarly, ebony feature is not produced together with normal antenna. And the reason being that linkage occurred and there were just no recombinant gametes. Now what have we done? We have taken male as the F1 offspring and female as the purebred recessive. Now just let's take another example of the same organism where the female F1 offspring was taken together with a male purebred recessive. So in this case, the female that we are taking is capital E, capital A, small a, and the male is small e, small a, small e, small a. Now, studies have shown that if a female is dihybrid, then this female will produce two type of separate type of gamete, which means four different gametes. In large quantities, the gametes will be the same as the parental class. In fewer quantities will be the recombinant gametes because the linkage group will break. This is a rare chance, but yes, this occurs. So four gametes will be produced, but they will not be in equal proportion. Now, when you cross them together, we have the same striped and normal and ebony histopedia being there in the pentaclass. As pentaclass, the F2 offspring also shows the same phenotype, but striped and histopedia and ebony normal also shows up as recombinants. We expect them to be in 1:1 ratio. We expect them to have in the 1:1 ratio, the recombinants being there, pentaclass being there. So is it 1:1 ratio? No, when we count them, we see this kind of an offspring ratio being produced. These are not 25% each. We see pentaclass being more and recombinant class being less. Now this is a worked example here. So the next thing is how do you calculate the crossover value? The crossover value in this case is calculated just by adding the recombinant classes. In one of the papers, they gave a crossover value formula. We'll be covering that question in one of the videos later on. So you can see that in my past videos of P4 as well. So this is the recombinant class. So crossover value, for example, if I had 6 by 6 here, my value comes out 12%. So what does crossover value shows? This is what I'm discussing. The crossover value is proportional to the distance between loci. For example, if my crossover value comes out to be 12%, it means that the loci E and A are very close together. The distance between these loci is very small. Okay, now what I would recommend for you to do is pick this question number one from this page, this paper, October November 2016, video 43. This question deals with epistasis, and this will get you a kind of cross an exposure of what kind of question can we get when we are discussing epistasis. Similarly, there's a question number seven on May June 42, 2017. Go for this question as well and try to see how the question is being attempted and try to do it yourself to see how this goes on. Then there's a question regarding the linkage group that I've just discussed. This is May June 2018 variant 42 question 2. This question deals with exactly the same kind of description that I've mentioned, and this is the question where they have given to you the crossover value formula that I was talking about. So that's it from my side. I hope this was helpful for you to follow the seven different type of crosses that we have covered in these two videos. I hope this was helpful for you. That's from my side. Thank you so much.